On this occasion, we're diving deep into gene editing, and specifically CRISPR.
Oh, very exciting.
Yeah,
It's such a fascinating field.
it really is. We'll be talking about the potential to revolutionize medicine, agriculture, but also the ethical
Yep,
concern.
it's a real double-edged sword, isn't it?
Yeah, it is. So we're going to explore the history of gene editing, the mechanisms behind CRISPR, its potential applications, and all the ongoing debates around it.
Looking forward to it.
So, you know, gene editing, it's had such an interesting evolution, right?
Absolutely.
Like going from, selective breeding to the precision of CRISPR.
Yeah, we've come a long way from just sort of picking, the best seeds or breeding animals for certain traits
Yeah, and it's interesting to think that, even back then, humans were already shaping the genetic makeup of organisms
without even realizing it.
without even really realizing
Yeah.
it. Yeah, exactly. And then came the recombinant DNA technology in the 1970s,
A huge turning point.
which was huge. It allowed scientists to directly manipulate DNA.
Yeah, it was revolutionary that we could actually transfer genes between different species.
Amazing.
Things like producing insulin in bacteria. I mean, it really opened up so many doors.
And that paved the way for CRISPR.
It did.
And so CRISPR is interesting because it comes from this natural defense mechanism in bacteria, right?
It does, yeah. It's like a bacterial immune system
Huh.
that remembers past viral encounters.
It's like a library,
It is like a library.
a genetic library.
Exactly. So it uses this system to identify and cut up the DNA of invading viruses.
So it can protect itself.
Exactly, from future infections.
So how does that turn into a tool for editing human genes?
Right. Well, scientists figured out that they could create a guide RNA,
Yep.
which is a molecule that acts like a GPS to target a specific DNA sequence.
Oh, wow.
And they paired this guide RNA with the Cas9 protein, which acts like scissors,
Molecular scissors.
molecular scissors, to cut the DNA at that location.
So it's like a find and replace function.
That's a great way to put it. Yeah.
But after it's cut, what happens next?
So the cell has its own natural repair mechanisms.
Okay.
And scientists can harness those mechanisms to either disrupt a gene, essentially turn it off, or they can introduce a new piece of DNA
Oh, wow.
and basically rewrite the genetic code.
Rewrite the blueprints of life.
That's right.
But I mean, with any powerful technology, we worry about the downsides, right?
Of course there are concerns.
The ethical concerns.
Absolutely. We're still learning so much about the genome.
Right.
Editing one gene could have unintended consequences
Unforeseen effects.
on other genes or the entire organ.
Yeah, a domino effect.
Exactly.
And I imagine the other fear is misuse, right?
Of course.
Like designer babies or, things that could exacerbate social inequality.
Right. We need to have very strict ethical guidelines and regulations to prevent harm to individuals or society.
And so these conversations, they need to involve not just scientists, but
Everyone.
ethicists, policymakers, the public.
It's got to be a group effort.
Yeah, definitely. We need as many perspectives as possible.
Absolutely.
And, CRISPR is constantly evolving, right? New techniques, applications.
All the time. Yeah,
It's a fast-moving field.
it really is.
You mentioned something earlier called prime editing.
Oh, yeah.
Can you tell me a little bit more about that?
So prime editing is a more precise form of gene editing.
Okay.
So unlike CRISPR-Cas9, which cuts both strands of the DNA,
Right.
prime editing only nicks one strand and uses an enzyme to directly write the new information into that target site.
So instead of scissors, we're using a pen,
Yeah, a very, very precise pen.
which reduces the risk of unintended edits.
Massively.
So the implications for this are huge.
Oh, they're massive. We could be looking at so many more diseases and conditions that we could target.
That's incredible.
An exciting time.
It is. So we've covered a lot, the history of gene editing, how CRISPR works, its potential applications, and the ethical concerns. And it feels like we're at the very beginning of this genetic revolution,
I think we are.
with CRISPR leading the charge.
It is, but we have to remember to proceed with caution
Yes.
and be really aware of the ethical and societal implications.
What are your thoughts on that?
I think it's the most important thing to consider. We have to use this technology responsibly.
For the benefit of humankind.
Absolutely. We need dialogue and collaboration between everyone involved.
Yeah, I agree.
Yeah, it's a really exciting time for CRISPR.
It is.
With so many advancements happening.
It does feel like we're on the cusp of something,
Absolutely.
like a new era. In medicine especially, I've been reading about how scientists are using CRISPR to cure genetic diseases like sickle cell anemia, cystic fibrosis. It's incredible to think we might actually be able to eradicate those diseases.
Yeah, and the potential goes even beyond treating diseases.
Really?
Think about food security.
Okay.
CRISPR can engineer crops that are resistant to, pests and diseases and even harsh environmental conditions.
That could be huge
It really could.
for agriculture and feeding a growing population.
Exactly.
But, I mean, we keep talking about this great power, right?
Right.
It becomes great responsibility. So what are the steps being taken to ensure that CRISPR is used ethically?
Well, the scientific community is having these discussions all the time
Okay.
about the ethics and the regulations of CRISPR research.
That's good.
It's not just about pushing the boundaries of science.
Right.
We have to make sure that it aligns with our values.
And it doesn't lead to unintended consequences.
Exactly.
Yeah, so we've talked about the benefits, we've talked about the concerns. What about the actual research? Like what's happening right now with CRISPR? Are there clinical trials? Are there real-world applications?
Yeah, there are actually quite a few clinical trials going on. Right now, they're using CRISPR to treat diseases like cancer, blood disorders, even inherited genetic conditions.
And are they seeing promising results?
Some early results have been really promising, which is great.
So it seems like it's moving from the lab to the real world
It is.
and bringing us closer to realizing the potential. Are there any specific breakthroughs or advancements in CRISPR technology that you're excited about?
Well, they're refining CRISPR-Cas9 all the time,
Okay.
making it more and more precise and efficient.
To reduce those off-target effects.
Yeah, minimize those risks as much as possible.
And we talked about prime editing before,
Oh, yeah.
which sounds like a game changer.
It is in terms of precision and versatility. So prime editing can make these really precise changes to the DNA sequence without cutting the DNA double helix.
Which the traditional CRISPR does.
Exactly. So it reduces those unintended genetic changes.
It sounds like we're moving towards this future where gene editing isn't just fixing broken genes, but enhancing,
Yeah.
fine-tuning human health and well-being.
Yeah, it's a really exciting but also daunting prospect.
It is.
We have to make sure we're using it ethically and equitably.
So this has been amazing. We've gone from the history of gene editing to the details of CRISPR and how it could impact our world.
It really is a revolutionary technology.
It is, and CRISPR is at the forefront of it all.
But with all this potential, we have to think about the ethics and the societal implications.
Yes.
And have these open discussions with diverse perspectives.
Definitely to make sure it benefits all of humankind.
Absolutely.
I want to talk for a moment about media coverage,
Yeah.
because we're constantly bombarded with information.
Yeah.
So how do we, as consumers of information, distinguish the accurate from the sensationalized?
Right. Well, look for sources that rely on scientific evidence.
Okay.
You want to avoid anything that feels like hype or fear-mongering.
Yeah.
And look for balanced perspectives, looking at both the potential benefits and the risks.
And remember that scientific progress is a journey. It's not a destination.
Absolutely.
There are going to be setbacks, challenges, and unexpected discoveries.
Always.
So we need to approach it with curiosity,
Yeah.
with open-mindedness, and a commitment to being ethical and responsible.
I think that's a perfect way to put it.
So now I want to talk about something that I think sparks a lot of debate, and that's the use of CRISPR in human embryos. That's a big one.
It is. It's a topic that's fraught with ethical and societal considerations, and that's what we'll explore in the next part of our deep dive.
The potential is pretty profound, isn't it?
It really is.
Yeah.
It makes you think about what it means to be human.
Yeah. Where do we draw the line?
Exactly.
Yeah.
On the one hand, we have the potential to prevent these terrible genetic diseases from being passed down.
That's right, to eliminate that suffering.
But then there's that other side,
Mm-hmm.
the unintended consequences,
Right.
the genetic discrimination, and the slippery slope to designer babies.
It's a big fear.
It is. And, one of the biggest concerns is introducing harmful mutations.
Into the gene pool.
Exactly.
Hmm.
And, mutations that could last for generations.
It's huge responsibility.
It is. Do we even have the right to do that? To make those kinds of changes?
It's a tough question.
It is. I don't think there's an easy answer.
Not at all. And I think it really needs those conversations,
Yeah.
between scientists, ethicists, policymakers, and the public. We need to figure out what's acceptable.
And where we draw that line?
Exactly. We can't ignore it.
No, we can't.
CRISPR is here. We have to use it responsibly. But it's hard to keep up, isn't it?
It's so fast moving.
Yeah, the science is advancing so quickly.
How do you regulate something that's constantly evolving?
Right. And there are so many different opinions.
Oh, absolutely. Some people think that any kind of germline editing is just wrong.
And then others think that we have a moral obligation to use this technology.
To ease suffering.
Yeah, it's so complex.
It really is. We just need to be cautious, humble, and respectful,
Of human life.
of human life, exactly.
So we've been on this journey, exploring gene editing and CRISPR.
It's been quite a deep dive.
It has, and we've uncovered so much potential, but also so many ethical considerations.
It really shows that we need to be informed and engaged,
Yeah.
We all have a responsibility to make sure these technologies are used for good.
To benefit everyone.
Exactly.
I'm walking away from this feeling both excited and a little bit overwhelmed.
I understand that.
CRISPR has this amazing potential, but it comes with such great responsibility.
It does. We need to make sure science is guided by ethics.
By a commitment to humanity and to our planet.
I couldn't have said it better myself.
So thank you for joining us on this deep dive.
We hope you learned something.
That it was thought-provoking, maybe even a little bit mind-blowing.
And we hope you keep exploring and questioning and engaging in these conversations that shape our world.
What are your thoughts on CRISPR and the ethical considerations we've discussed? Let us know. We'd love to hear from you.